There was an era that lasted 287 million years, nearly twice as long as the age of dinosaurs and the age of mammals combined, and about which most people know almost nothing. The Paleozoic Era. It began when life first learned to have eyes. It created the first forests, the first bones, the first claws. It produced creatures the size of a school bus that swam in the ocean, and dragonflies with wingspans like an eagle that patrolled the forests. It nearly destroyed all life twice. And at its very end, just a few million years before the dinosaurs appeared, it created animals so close to mammals that the difference was only a matter of a few bones in the skull. This is their story. Before we go any further, please subscribe to this channel. Your support really helps us to continue this work. Thank you. There is a number that puts the Paleozoic era in perspective, and it is not the 287 million years of its duration, impressive as that is. The number is 539. As in 539 million years ago, the moment when the Cambrian period began and the Paleozoic era opened. To understand why that number matters, you need to understand what the world looked like the day before it. Because the world before the Cambrian was, by any measure of biological complexity, almost nothing. For roughly 3 billion years before the Cambrian, life on Earth had been almost exclusively microbial. Bacteria, archaea, and eventually single-celled eukaryotes dominated the oceans. The Ediacaran fauna that appeared after the snowball Earth events of the Cryogenian, those strange, quilted and frond-like organisms we have discussed in previous episodes, represented the first experiments in multicellular complexity, but they were experiments that left almost no descendants. They were soft, slow, largely immobile, feeding on organic matter in the sediment or filtering nutrients from the water. The world before the Cambrian was quiet in a way that is difficult to imagine. No predators, no shells, no bones, no eyes, no pursuit, no escape, no arms race between hunter and hunted. The ocean floor was a microbial mat punctuated by the occasional Ediacaran frond, and that was essentially it. And then, in an interval of geological time so brief that it barely registers on a 4.5 billion year timescale, everything changed. The Cambrian Explosion. The appearance of virtually all major animal body plans within roughly 20 million years, beginning approximately 539 million years ago, is one of the most dramatic and most debated events in the history of life. It is dramatic because the contrast between what came before and what came after is so stark. It is debated because the cause of that contrast has never been fully resolved, and the debate has been ongoing for more than a century without producing consensus. What is not debated is the reality of the explosion itself. The fossil record is clear. Before the Cambrian, animal diversity was minimal. After the Cambrian, the full range of animal body plans that would characterize every subsequent chapter of evolutionary history was present. The trigger for the Cambrian explosion is likely a combination of factors that intersected at approximately the same geological moment. The oxygenation of the deep ocean, which had been occurring gradually through the late Proterozoic as atmospheric oxygen levels rose following the Great Oxidation Event, finally reached levels sufficient to support the aerobic metabolism that complex animal tissue requires. The chemical richness of the post-snowball Earth-ocean, which we discussed in our episode on ice ages, had primed the biological stage. And perhaps most importantly, the evolution of predation. The appearance of animals that actively hunted and consumed other animals initiated the evolutionary arms race that would drive the rapid diversification of defensive structures, sensory organs, and locomotor systems that characterizes the Cambrian fauna. The appearance of eyes in the Cambrian is the key. Not merely the evolution of light-sensitive cells, which had existed in various forms for hundreds of millions of years before the Cambrian, but the evolution of sophisticated image-forming eyes capable of detecting the position, size, and movement of other organisms at a distance. The compound eye of the trilobite, which appears in the fossil record at the very beginning of the Cambrian, and which is one of the most sophisticated optical instruments in the history of life, with hundreds or thousands of individual lenses creating a wide-angle image of the surrounding environment, represents a technological leap whose consequences for the biology of the entire planet were immediate and permanent. Once one animal can see, everything changes. The animals it can see now face selection pressure to avoid being seen, to develop camouflage, armor, warning coloration, or the ability to hide in the sediment. The animals that develop armor now face selection pressure in their predators towards stronger jaws, sharper claws, faster pursuit. The arms race escalates, and every escalation drives new evolutionary innovation in both predators and prey. This is why the Cambrian explosion produced body plans so rapidly, not because evolution was operating faster than normal, but because the selective pressures of the predator-prey arms race were creating new niches and new challenges faster than at any previous point in the history of animal life. The most famous windows into the Cambrian world are the exceptional fossil preservation sites called Lagerstätten, German for storage places, where the soft tissues of Cambrian organisms were preserved in unusual geological conditions that prevented their decay before burial. The Burgess Shale of British Columbia, discovered by the American paleontologist Charles Walcott in 1909, is the most celebrated of these sites. Its fossils, preserved in fine-grained black shale, dating to approximately 508 million years ago, reveal a fauna of extraordinary diversity and strangeness. Animals that fit into no modern category and that have sparked decades of debate about their relationships to living animal groups. Anomalocaris, the largest predator of the Cambrian seas, was a creature that defied easy classification when it was first described. Its fossils were initially found in pieces and misidentified. The circular mouth was described as a jellyfish. The grasping appendages were described as shrimp tails, and the body was described as a sponge. Only when complete specimens were eventually found did the full animal become clear. Anomalocaris reached lengths of perhaps 1 meter, about 3 feet, enormous by Cambrian standards, with 2 large compound eyes on stalks, 2 segmented grasping appendages at the front of its head for seizing prey, a circular mouth with radiating plates for crushing hard-shelled organisms, and undulating side lobes for propulsion through the water. It belonged to the broad group that would eventually produce all modern arthropods: insects, crustaceans, spiders, and their relatives. But it was not closely related to any specific modern group. It was an early experiment in the arthropod body plan, extraordinarily successful for its time and then gone, leaving no descendants. Hallucigenia, named for the hallucinatory quality of its appearance, was a creature that paleontologists initially reconstructed upside down. It walked on paired spines that were initially interpreted as legs, with tentacle-like legs that were initially interpreted as dorsal appendages. When the correct orientation was finally determined in the 1990s, the animal resolved into something only marginally less strange than the original reconstruction. A small worm-like creature with 7 or 8 pairs of walking legs on its underside, a corresponding row of pairs of tentacles on its back, and a head with a pair of eyes and a circular mouth ringed with teeth. Hallucigenia belongs to a group called the velvet worms, whose living relatives, soft-bodied, caterpillar-like animals found in tropical forests today, are among the most ancient of living animal lineages, survivors from the Cambrian that have changed remarkably little in 500 million years. Opabinia, with 5 eyes and a flexible proboscis ending in a grasping claw, was so strange when first formally described at a scientific meeting in 1972 that the audience reportedly laughed. It has since been recognized as a genuine animal and placed as a relative of the anomalocaridids. But its 5-eyed face remains one of the most arresting images in all of paleontology. The Cambrian was a world of biological experimentation on a scale that has not been matched since, and the experiments were not constrained by the need to fit into any pre-existing ecological framework. Every niche was open. Every solution to the problem of surviving in a complex, predator-rich ocean was worth trying. The Cambrian fossil record has a second extraordinary window that rivals the Burgess Shale in its importance and surpasses it in the completeness of its preservation. The Chengjiang biota of Yunnan province in southwestern China, discovered in 1984 by the Chinese paleontologist Hao Xiangwang and dating to approximately 518 million years ago, slightly older than the Burgess Shale, preserves a Cambrian fauna of even greater diversity and 3-dimensional fidelity than its Canadian counterpart. The Chengjiang fossils include not only the external form of Cambrian animals, but their internal organs: the gut, the nervous system, the musculature, preserved in sufficient detail to allow researchers to reconstruct the physiology and behavior of animals that died over 500 million years ago with a precision that was unimaginable before these fossils were studied. Among the Chengjiang fauna is Picaia gracilens, or its Chengjiang relative Unanozune lividum, the earliest known chordate, the distant ancestor of every vertebrate that has ever lived, including you. It was a small blade-like swimming animal, roughly 5 centimeters, about 2 inches, long, with a notochord, the stiffening rod that is the precursor to the vertebral column, running the length of its body and V-shaped muscle blocks on either side that it used for swimming. It was not impressive. Nothing in its appearance would have suggested that its descendants would eventually include sharks, dinosaurs, and humans. But the notochord was the architectural prerequisite for the vertebrate body plan, and Pikaia was carrying it through the Cambrian seas at a time when everything else in the ocean was an invertebrate. The Cambrian also witnessed the appearance of the first mineralized shells, a development of such importance that it deserves its own explanation. Before the Cambrian, organisms were soft-bodied. The evolution of the ability to precipitate calcium carbonate or calcium phosphate into hard structures, shells, teeth, spines, claws, happened independently in multiple lineages at approximately the same time in the earliest Cambrian. This simultaneous appearance of biomineralization in unrelated groups is one of the most striking features of the Cambrian explosion, and one of the strongest arguments that the explosion was driven by a systemic environmental change rather than by independent evolutionary innovations in each lineage. The most likely explanation for the simultaneous appearance of biomineralization is the same as the explanation for the simultaneous appearance of predation. They are cause and effect. Once animals began hunting other animals, the selection pressure for protective hard parts became intense in every prey lineage simultaneously. The calcium and phosphate ions needed to build shells and teeth were available in the ocean. They had been present throughout the Proterozoic, but had not been exploited for structural purposes because there was no selection pressure to do so. Once predation began, the selection pressure appeared simultaneously in every prey group, and biomineralization evolved independently in dozens of lineages within a geologically brief interval. The small shelly fauna, the assemblage of tiny, isolated mineralized pieces that appear at the very base of the Cambrian in rocks worldwide, represents the first wave of this biomineralization event. Before the Burgess Shale animals appeared, the Cambrian seas were already populated by creatures whose external skeletons broke up into thousands of tiny plates and spines and tubes. When the animal died and the tissues holding the pieces together decayed. Reconstructing the original animals from these disarticulated fragments has been one of the great challenges of Cambrian paleontology, and the solutions, achieved through careful examination of specimens where the skeleton remained partially articulated, have revealed a diversity of early skeletal animals that preceded the familiar Burgess Shale fauna. The trilobites deserve particular attention, both because they are the most abundant and diverse animals in the Cambrian fossil record, and because their history spans the entire Paleozoic era, making them the most enduring success story of the era from beginning to end. Trilobites, named for the three-lobed body plan visible in their fossilized exoskeletons, were marine arthropods with a segmented body protected by a mineralized exoskeleton that molted as the animal grew, leaving behind a fossil record of extraordinary completeness. They first appear at the very beginning of the Cambrian, 538 million years ago, and they persist until the Permian extinction, 252 million years ago, a run of nearly 300 million years that exceeds the current reign of mammals by more than 4 times. In their 300 million year history, trilobites diversified into more than 20,000 described species, occupying virtually every marine ecological niche available to a bottom-dwelling or pelagic arthropod. Some were predators, equipped with strong spines and large compound eyes for tracking prey. Some were filter feeders, with elaborate gill-bearing appendages for extracting organic particles from the water. Some burrowed in the sediment, their eyes reduced or absent. Some rolled into a defensive ball when threatened, exactly as pillbugs do today, which are, not coincidentally, also arthropods, descendants of the same great group that produced the trilobites. The trilobite compound eye is one of the oldest sophisticated visual systems in the fossil record. And some trilobite species evolved eyes of extraordinary optical precision, with individual lenses composed of calcite crystals, oriented so as to minimize optical aberration, a solution independently arrived at by human lens makers only in the 17th century. The Cambrian world was primarily an ocean world. The land was bare rock and sand, colonized only by microbial crusts and perhaps the very earliest photosynthetic algae. There were no plants in the terrestrial sense, no roots, no stems, no leaves, no vascular tissue to transport water against gravity. There were no terrestrial animals of any kind. The entire drama of the Cambrian Explosion played out in the sea, and the sea was the only theatre of biological innovation for much of the subsequent Ordovician period as well. The Ordovician period, spanning from 485 to 444 million years ago, is in some ways the most underappreciated chapter of the Paleozoic. It lacks the dramatic narrative hooks of the Cambrian explosion that preceded it, or the colonization of land that began in the Silurian that followed it. But in terms of sheer biological diversity, the Ordovician was extraordinary. The Great Ordovician Biodiversification Event, the most rapid and extensive increase in marine animal diversity in the entire Paleozoic record, roughly tripled the number of marine animal genera between the early and late Ordovician. The shallow tropical seas of the Ordovician world, warmed by a climate that had no ice sheets through most of the period, supported reef communities of extraordinary complexity, dominated by tabulate and rugose corals, stromatoporoid sponges, and the most elaborate crinoid communities the world has ever seen. Crinoids, the sea lilies, which are echinoderms related to starfish and sea urchins, reached their greatest diversity and ecological importance in the Ordovician and Silurian. They are filter feeders, anchored to the seafloor by a stalk and extending a crown of feather-like arms to capture organic particles from the water. In the shallow Ordovician and Silurian seas, crinoid meadows covered the seafloor in such density that the accumulated remains of their calcite skeletons produced thick limestone deposits. The crinoid limestones that today provide the building stone of many historic structures in the American Midwest and the British Isles. Walking through a quarry in Indiana or through the streets of a town built from local limestone is, in a very literal sense, walking through an Ordovician crinoid meadow. But the Ordovician also produced a group of animals that has haunted the imaginations of everyone who has encountered their fossils: the Eurypterids. These were chelicerates, relatives of the horseshoe crabs and the ancestors of the arachnids, that occupied the predatory niche in both marine and freshwater Paleozoic environments, from the Ordovician through the end of the Permian. The largest Eurypterids, which appeared in the Silurian, reached lengths of 2.5 meters, about 8 feet, making Echolopterus rhenanii, known from a claw fragment found in Germany, the largest arthropod that has ever lived. They were marine scorpions in the broadest sense, not true scorpions, which evolved later from terrestrial relatives, but scorpion-like predators with a segmented body, a pair of large compound eyes, multiple pairs of walking legs, and in many species a telson, a tail spine, that may have served as both a defensive weapon and a sensory organ. Eurypterids appear to have been among the dominant large predators of the Silurian and Devonian oceans and freshwater systems, hunting fish and other invertebrates with their raptorial front claws and pursuing prey through the water with their paddle-like hind limbs. Their fossils are found on every continent, including Antarctica, and they persisted for over 200 million years before going extinct at the end of the Permian, along with most of the other characteristic Paleozoic marine fauna. The fact that they are not household names in the way that dinosaurs are reflects nothing about their biological significance and everything about the popular appetite for terrestrial giants over marine ones. The Ordovician seas were also home to the nautiloids, the ancestors of the modern nautilus and the distant relatives of the squid and octopus. And in the Ordovician and Silurian, they were among the most formidable predators in the ocean. The orthoconic nautiloids, those with straight rather than coiled shells, reached lengths of up to 9 meters, about 30 feet, in some Ordovician species, making Cambrian the largest animal known to have lived in the Ordovician seas. These straight-shelled cephalopods hunted the seafloor communities of trilobites, brachiopods and crinoids with a speed and agility that the sessile or slow-moving invertebrates of the Ordovician Ocean were ill-equipped to escape. Their shells were internally divided into chambers that the animal could fill with gas or liquid to control its buoyancy, allowing them to hover at any depth in the water column and descend rapidly to the seafloor to strike at prey. The graptolites deserve mention as one of the most ecologically significant groups of the early Paleozoic Ocean, even though they are almost never mentioned in popular accounts of ancient life. Graptolites were colonial animals, each colony consisting of dozens to hundreds of individual organisms called zooids, housed in a series of cups along a branching or simple stem. They were planktonic, drifting in the surface waters of the ocean and filtering organic particles from the water. Their colonies, which fossilized as distinctive saw blade or double saw blade shapes in black shales worldwide, are among the most useful guide fossils in stratigraphy because they evolved rapidly and are found in enormous quantities in Ordovician and Silurian rocks. They went through multiple rapid evolutionary radiations during the Ordovician, producing hundreds of species whose stratigraphic ranges allow geologists to correlate rocks of the same age on different continents, with a precision of hundreds of thousands of years. The Silurian brought the most consequential vertebrate innovation of the early Paleozoic, the evolution of jaws. The earliest vertebrates, the jawless fish or agnathans, had mouths but no jaws. They fed by filter feeding, by suction, or by rasping at surfaces with specialized mouthparts. But they could not bite. The evolution of jaws from the anterior gill arches, the bones that support the gill slits behind the head, transformed the vertebrates from passive or opportunistic feeders into active predators capable of seizing and processing a far wider range of prey. The first jawed fish appear in the Silurian, and their evolutionary radiation in the Devonian produced the extraordinary diversity of armored fish and sharks that dominated the Devonian seas. The Silurian also produced the first scorpions, true terrestrial scorpions, descended from aquatic arachnid ancestors, making their way onto land and becoming among the earliest terrestrial predators. The scorpion body plan, with its pincers, segmented tail, and venomous sting, appears in the Silurian fossil record in a form almost identical to modern scorpions. Like the coelacanth and the horseshoe crab, the scorpion has changed so little in 440 million years that it can reasonably be called a living fossil, a body plan so well adapted to its ecological role that the evolutionary pressure to change has been minimal. The first mass extinction of the Paleozoic, and the second largest in the history of animal life, struck at the end of the Ordovician, approximately 445 million years ago. The Hernantian glaciation, which we discussed in our episode on supercontinents in the context of Gondwana passing over the South Pole, drove a massive drop in sea levels as water was locked up in the growing continental ice sheets. The tropical and subtropical shallow marine environments that had supported the extraordinary Ordovician biodiversity were drained. Ocean temperatures plummeted. The chemical gradients that the warm Ordovician seas had maintained shifted dramatically. Approximately 85% of marine species went extinct. The trilobites were dramatically reduced, the graptolites were nearly eliminated, the crinoid and coral reef communities collapsed. The world that emerged from the Ordovician extinction was a diminished version of the rich Ordovician biosphere, and the recovery took millions of years. The Silurian period, 444 to 419 million years ago, is in some ways the recovery chapter of the early Paleozoic, but it is also the chapter in which one of the most consequential events in the history of life on this planet occurred: the colonization of the land. The land had not been entirely empty before the Silurian. Microbial mats had colonized terrestrial surfaces since the Archean. There is evidence of soil formation and simple surface communities in the Ordovician and possibly earlier. But the Silurian is when the first vascular plants with an internal system of tubes for conducting water and nutrients, the biological equivalent of a plumbing system, are documented in the fossil record. Before vascular plants, the colonization of the land by photosynthetic organisms was limited to thin surface crusts of algae, cyanobacteria, and fungi, none of which could grow to any significant height, because without a vascular system, water could only be transported by diffusion over very short distances. The evolution of vascular tissue, the xylem for transporting water upward from the roots, and the phloem for transporting sugars downward from the leaves, made it possible for plants to grow tall, to extend roots deep into the soil, and to occupy the interior of continents far from the water's edge. The earliest vascular plants of the Silurian, Cooksonia and its relatives, were small, leafless, and simple by the standards of what would follow. They were essentially upright green stems with branching tips, reproducing by spores, growing in the damp soils of river floodplains and coastal environments. They had no leaves, no roots in the modern sense, and no secondary wood. But they had the vascular system that was the architectural prerequisite for everything that came after. They were the prototype from which 350 million years of plant evolution would eventually produce the flowering plants, the conifers, the ferns, and the mosses that cover most of the terrestrial surface of the modern Earth. The arthropods were close behind. The first evidence of terrestrial arthropods, millipede-like animals and possibly early arachnids, comes from the Silurian, though the fossil record is fragmentary, and the question of exactly when the first arthropods colonized the land permanently is still debated. What is clear is that by the early Devonian, both plants and arthropods were well established on land, and the stage was set for the most dramatic chapter of Paleozoic terrestrial evolution. The Devonian period, 419 to 359 million years ago, is called the Age of Fishes, and the name is fully deserved. The Devonian oceans and freshwater systems were dominated by fish in a diversity and ecological richness that has never been equaled in the subsequent history of vertebrate aquatic life. But the Devonian is also the period in which the vertebrates first conquered the land, and that event, the emergence of the first tetrapods from the Devonian water, is one of the most consequential evolutionary transitions in the entire history of complex animal life. The fish of the Devonian were not the fish we know today. The dominant groups of the Devonian seas were the placoderms, the armored fish, and the acanthodians, and the vast majority of them have no living descendants. The placoderms were encased in bony armor covering their heads and the front of their bodies, with the back half of the body covered in scales or naked skin. They had hinged jaws, one of the critical innovations of early vertebrate evolution, because jaws allow a far wider range of prey than the filter feeding or suction feeding strategies available to jawless fish. And they occupied every ecological niche from small bottom-feeding detritivores to the apex predators of the Devonian seas. The apex predator of the Devonian Ocean deserves particular attention. Dunkleosteus terrelli was a placoderm that reached lengths of 8 to 9 meters, about 26 to 30 feet, the largest vertebrate predator the world had yet seen. Its head and anterior trunk were encased in interlocking bony plates. But instead of teeth in the conventional sense, Dunkleosteus had shearing blades of bone, self-sharpening edges created by the differential wear of harder and softer bone layers that could generate a bite force estimated at somewhere between 5,000 and 6,000 newtons. That is roughly comparable to the estimated bite force of a large crocodile, concentrated in blades rather than individual teeth capable of cutting through the armor of other placoderms. Dunkleosteus was not hunting soft prey. It was hunting other armored fish. The Devonian Ocean was an arms race between ever thicker armor and ever more powerful jaws. The sharks also appear in the Devonian fossil record, though their early history is poorly known because cartilaginous skeletons rarely fossilize. By the late Devonian, sharks were already diversifying into the ecological roles they would occupy for the next 400 million years. Fast-swimming open-water predators, slower bottom-feeding sharks, and specialized forms with flattened, crushing teeth for feeding on hard-shelled invertebrates. The shark's combination of sensory sophistication, the lateral line system for detecting water vibrations, the ampullae of Lorenzini for detecting electrical fields, and acute smell, with hydrodynamic efficiency, has made them one of the most enduring body plans in the history of vertebrate life. But the lobe-finned fish, the Sarcopterygians, are the group that matters most for the subsequent history of life, because it is from within this group that the first four-limbed vertebrates evolved. Lobe-finned fish have fleshy, muscular fins supported by an internal skeleton of bones that are homologous to the bones of the tetrapod limb. The humerus, radius, and ulna of the forelimb, the femur, tibia, and fibula of the hind limb. In modern lobe-finned fish, represented by the lungfish and the coelacanth, these fins are used for slow, deliberate locomotion along the bottom of rivers and lakes. In the Devonian, the lobe-finned fish were far more diverse, occupying a range of ecological niches in both marine and freshwater environments. The transition from lobe-finned fish to four-limbed tetrapods is documented in the fossil record with a completeness and resolution that was not available even 30 years ago. The discovery of Tiktaalik rosei in 2004 in the Canadian Arctic by a team led by Neil Shubin of the University of Chicago provided the clearest transitional form yet found between fish and tetrapods. Tiktaalik lived approximately 375 million years ago in what were then warm, shallow rivers and swamps in tropical North America. Its body was fish-like, with scales, gills, and fins, but its head was flat and its neck was mobile, both features unknown in fish, which typically have their head fused to their body. Its pectoral fins contained a shoulder, elbow, and wrist, the full complement of joints that allows a tetrapod to push itself up from the ground. It could almost certainly prop itself up and look around in a way that no conventional fish could. Shubin has described Tiktaalik as a fish that could do push-ups. Why did the vertebrates leave the water? The traditional answer, that the drying of shallow Devonian pools forced fish to crawl between water bodies, has been largely abandoned by modern researchers, who note that the locomotor adaptations of early tetrapods are more consistent with animals that moved through shallow water than with animals that crawled on dry land. The current consensus is that the evolution of terrestrial locomotion began as an adaptation for moving through dense aquatic vegetation in shallow water, and that terrestrial locomotion was a secondary consequence of adaptations that were initially selected for aquatic performance. The land was not the destination. It was the accidental beneficiary of an adaptation that had evolved for a different purpose. What the land offered, once animals arrived, was ecological opportunity of the same kind that the Cambrian Explosion had offered to the first complex multicellular animals. Empty niches, absent predators, abundant food. The terrestrial plant communities of the late Devonian, which had diversified dramatically from the simple Silurian stems into the first true trees. With woody secondary growth and broad leaves, provided both food and habitat for the first terrestrial vertebrates. The arthropods that had been on land since the Silurian were available as prey. The soils that the plants had developed were rich in organic matter and invertebrates. The first tetrapods that ventured onto land did not find a hostile environment. they found a world that had been waiting for them. The Devonian fish diversity extended far beyond the placoderms and the sharks. The actinopterygians, the ray-finned fish, whose descendants include virtually every modern bony fish from goldfish to tuna, first appear in the Devonian, initially as minor components of fish communities dominated by the more spectacular armored groups. They would not come to ecological dominance until the Mesozoic, when the extinction of the placoderms and many other Paleozoic fish groups opened the niche space that the rayfins subsequently filled. But the Devonian fossil record already shows the basic body plan. The fan of fin rays supported by a bony internal skeleton, the operculum covering the gills, the swim bladder for buoyancy control, that would eventually produce the most species-rich group of vertebrates alive today. The Cleveland Shale of northeastern Ohio, a black shale deposited in the deep anoxic waters of an inland sea that covered much of North America in the late Devonian, is one of the most productive sources of Devonian fish fossils in the world and has produced some of the most spectacular specimens of Dunkleosteus and its relatives. The black shale environment preserved these animals because the anoxic bottom water prevented scavengers and bacteria from consuming the carcasses before burial. The Cleveland shalefish died, sank to the anoxic bottom, and were preserved in extraordinary three-dimensional detail by rapid burial in the fine sediment of the deep basin. Dunkleosteus specimens from the Cleveland Shale show not only the bony armor of the head and trunk, but evidence of the bite force that made this animal the apex predator of its world. Bite marks on the armor of other placoderms, prey that had been bitten in half. Even regurgitated pellets of indigestible fish armor that Dunkleosteus apparently produced after digesting the soft parts of its prey, exactly as modern raptors produce pellets of fur and bone. The transition from water to land in the Devonian was not a single event, but a process that unfolded over millions of years and involved multiple lineages of lobe-finned fish independently experimenting with amphibious locomotion. The Devonian lobe fins that preceded Tiktaalik include Panderichthys, a flat-headed fish with a mobile neck and pectoral fins, whose internal skeleton already showed the tetrapod limb pattern, and Elpistostege, discovered in Quebec in 2010 and described in detail in 2020, which preserved the most complete record yet found of the fin-to-limb transition. Elpistostege had all the bones of the tetrapod hand, the radius, ulna, and the beginnings of finger-like elements, arranged in a configuration that suggests it was capable of weight-bearing on the bottom of shallow water environments. The sequence from Panderichthys to Elpistostege to Tiktaalik to the first true tetrapods, Acanthostega and Ichthyostega from Greenland, is one of the most complete evolutionary sequences of a major body plan transition in the entire fossil record. Acanthostega had 8 fingers on each forelimb, more than the 5 that would become standard for all subsequent tetrapods, and hind limbs that were almost certainly non-functional for walking on land. It was, in the current scientific consensus, a fully aquatic animal that had evolved limbs for locomotion through dense aquatic vegetation rather than for walking on land. The limbs came first. The land followed later as an accidental extension of a swimming adaptation. The late Devonian also produced the first forests. Archaeopteryx, the first true tree with a woody trunk, broad leaves and a crown structure recognizable as tree-like to modern eyes, appeared in the Middle Devonian and spread rapidly across the land surfaces of the late Devonian world, forming the first genuine forests. Archaeopteryx was not related to any modern group of trees. It was a fern-like plant that reproduced by spores rather than seeds. But it was architecturally a tree, and the forests it formed changed the chemistry of the planet in ways that had immediate and dramatic consequences for the global climate and for the ocean ecosystems that depended on that climate. Specifically, the forests of the late Devonian drove a massive drawdown of atmospheric CO2. The process by which this happened is the same silicate weathering process that drives CO2 drawdown whenever large areas of continental rock are exposed to the atmosphere, but the forests amplified it dramatically. Tree roots penetrate much deeper into rock than microbial mats or simple non-vascular plants, breaking up rock surfaces and exposing fresh mineral surfaces to weathering reactions. The organic acids that roots release accelerate the chemical weathering of silicate minerals. And the vast quantities of organic carbon stored in the woody tissue of forests represent carbon removed from the atmospheric CO2 pool and temporarily stored in biological form. The result was a dramatic decline in atmospheric CO2 through the late Devonian, a weakening of the greenhouse effect, and a cooling of global temperatures. This cooling contributed directly to the second mass extinction of the Paleozoic, the late Devonian extinction, which unfolded in a series of pulses between approximately 375 and 360 million years ago, and eliminated approximately 70% of all species. The late Devonian extinction was primarily a marine event. The reef communities of the Devonian, the rich coral and stromatoporoid reef ecosystems that had recovered from the end-Ordovician extinction and diversified through the Silurian and Devonian, were almost completely eliminated. The placoderms, which had dominated the Devonian oceans, were wiped out entirely. The diverse fish communities of the Devonian seas were drastically reduced. New research published in 2026 by Raki and Pisarskaya has reinforced the hypothesis that cataclysmic volcanism, specifically massive flood basalt eruptions in what is now Siberia, was the primary driver of the late Devonian extinctions, rather than the asteroid impact that some researchers had proposed. The volcanic emissions of CO2 and toxic metals appear to have disrupted ocean chemistry and triggered the ocean anoxia, the depletion of dissolved oxygen from deep ocean water. That is the primary kill mechanism documented in the late Devonian sedimentary record. The combination of cooling from the expanding forests and volcanic heating from the flood basalts created a climate system in unstable oscillation, and the ocean ecosystems caught in the middle of those oscillations paid the price. What survived the late Devonian extinction was a diminished but consequential fauna. The sharks survived and would diversify dramatically in the subsequent Carboniferous. The lobe-finned fish survived in their transitional forms, and the tetrapods they were producing continued their gradual conquest of the terrestrial environment. And the plants, the very agents that had helped trigger the extinction by drawing down CO2, survived and continued their extraordinary diversification onto land. The stage was set for the most visually spectacular and biologically extreme chapter of the Paleozoic. The Carboniferous period, 359 to 299 million years ago, gets its name from carbon, and the name is more literally accurate than it might appear. The vast coal deposits that powered the Industrial Revolution, that heated the homes of 19th century Europe and America, that fueled the steam engines that built the modern world. Those deposits are the compressed and fossilized remains of the Carboniferous forests. When you burn coal, you are releasing carbon that was fixed from atmospheric CO2 by Carboniferous trees 300 million years ago. The Industrial Revolution was, in a very direct geological sense, running on ancient sunlight stored in ancient wood. The Carboniferous world was dominated by a set of ecological and chemical conditions that have never been reproduced before or since. Atmospheric oxygen reached approximately 35%, nearly twice its modern level of 21%, driven by the extraordinary productivity of the coal forests and the failure of decomposition to keep pace with organic carbon production. The story of why oxygen was so high in the Carboniferous is inseparable from the story of lignin. Lignin is the structural polymer that gives wood its strength and rigidity. It is one of the most abundant organic polymers on Earth today, and it is produced by virtually all land plants as the primary structural material of their stems and trunks. But lignin is also one of the most chemically stable organic molecules in biology. It is extraordinarily resistant to decomposition, which is why dead trees rot so much more slowly than dead leaves. In the Carboniferous, lignin was a new molecule. The plants that produced it had evolved the biochemical pathway for lignin synthesis, as wood evolved in the late Devonian and early Carboniferous. But the microorganisms, specifically the fungi that decompose lignin in modern ecosystems, had not yet evolved the specific enzymatic machinery needed to break down this new molecule efficiently. The result was that Carboniferous trees fell, accumulated in the swampy lowland environments where they grew, and were buried by sediment faster than they could be decomposed. Carbon that would normally be returned to the atmosphere through decomposition was instead being buried, removing it from the carbon cycle. The carbon stayed in the ground. The oxygen stayed in the air, and the biology responded accordingly. The biological consequences of 35% atmospheric oxygen were immediate and dramatic. Oxygen concentration is one of the primary physical constraints on the body size of insects and other arthropods that breathe through passive diffusion rather than active pumping. The tracheal system of insects, the network of air tubes that delivers oxygen directly to the tissues, relies on diffusion to move oxygen from the atmosphere to the muscles and other oxygen-consuming tissues. The efficiency of diffusion decreases with distance, which means that in a modern 21% oxygen atmosphere, insects are limited to relatively small body sizes because oxygen cannot be delivered to the center of a large body fast enough by diffusion alone. But in a 35% oxygen atmosphere, Diffusion is so much more efficient that insects can grow to far larger sizes before oxygen delivery becomes limiting. The Carboniferous arthropods responded to this chemical gift with biological excess. Meganeura monyi, the giant Carboniferous dragonfly, had a wingspan of approximately 70 centimeters, about 28 inches, comparable to a modern crow or small hawk. It was not merely a large dragonfly. It was a fundamentally different kind of flying predator, occupying an aerial hunting niche that has not been occupied by an arthropod since. Arthropleura, the giant Carboniferous millipede, reached lengths of 2.5 meters, about 8 feet, and widths of 50 centimeters, about 20 inches, making it the largest land arthropod in Earth's history. It left distinctive trackways in Carboniferous coal measures across Europe and North America, 2 parallel lines of leg impressions separated by the central impression of the wide body, preserved in the fine-grained sediment of the Carboniferous swamp floors. The forests in which these giant arthropods lived were themselves extraordinary. The dominant trees of the Carboniferous coal forests were not the conifers or flowering trees that dominate modern forests. They were lycopsids, club mosses, that had evolved tree-like growth forms with woody trunks reaching heights of 30 to 50 meters, about 100 to 160 feet, and diameters of 2 meters, about 6.5 feet or more. Lepidodendron and Sigillaria, the most characteristic lycopsid trees of the Carboniferous coal forests, had bark covered in distinctive diamond-shaped leaf scars arranged in spiral patterns that give them an appearance unlike any modern tree. They grew in dense stands in the tropical swamps of Euramerica, the landmass formed by the collision of North America with Europe, and their fallen trunks and compressed leaf litter form the coal seams that are mined across the American Appalachian coalfields, the Pennsylvania anthracite region, the Yorkshire coalfields of England, and the Ruhr Valley of Germany. But the Carboniferous was not only a world of forests and giant insects. It was also the world in which the vertebrates took the final step that separated them from their dependence on water for reproduction. The amphibians, the first tetrapods, were successful and diverse in the Carboniferous, but they faced a fundamental biological limitation. Like modern frogs and salamanders, they had to return to water to reproduce. Their eggs, lacking the waterproof coating of a shell, would desiccate if laid on dry land. Their larvae required aquatic environments for development. This limitation kept the amphibians tied to the margins of water bodies and prevented them from colonizing the dry interior of continents. The solution to this problem, one of the most consequential evolutionary innovations of the Paleozoic, was the amniotic egg. The amniotic egg is a self-contained aquatic environment, a package that provides water, nutrients, and oxygen to the developing embryo while preventing desiccation and protecting against physical damage. It consists of the embryo surrounded by multiple membrane layers, the amnion, chorion, and allantois, that collectively regulate water exchange, gas exchange, and waste disposal, all enclosed in a leathery or calcified shell that prevents water loss while allowing oxygen and CO2 to diffuse. The first animals to lay amniotic eggs, could deposit them anywhere on dry land, free from the requirement to return to water for reproduction. The amniotic egg appears in the fossil record in the early Carboniferous, approximately 312 million years ago, with the first known amniotes, the ancestors of all reptiles, birds, and mammals. The exact animal that first laid an amniotic egg is not known because soft-shelled eggs do not fossilize readily. But the skeletal anatomy of the earliest known amniotes, like Hylonomus lyelli from the coal forests of Nova Scotia, shows the distinctive features of the amniote skull and skeleton that distinguish this group from the amphibians. Hylonomus was a small lizard-like animal, roughly 20 centimeters, about 8 inches, long, that lived among the hollow trunks of fallen lepidodendron trees in the Carboniferous coal forest. It was not impressive, but it carried within it the reproductive technology that would eventually free the vertebrates entirely from their aquatic origins and allow them to colonize the full range of terrestrial environments on Earth. The Carboniferous also witnessed a brief but consequential ecological crisis that is now recognized as one of the most important events in terrestrial evolutionary history. Romer's Gap. Named for the American paleontologist Alfred Romer, who first noticed it in the 1950s, Romer's Gap is an interval of approximately 15 million years in the early Carboniferous, from approximately 360 to 345 million years ago, during which the fossil record of terrestrial vertebrates is extraordinarily sparse. After the diverse tetrapod fauna of the late Devonian, the early Carboniferous record is nearly empty. A few fragmentary fossils suggest that tetrapods persisted through this interval, but the rich and diverse communities that existed before and after it are absent. The most widely accepted explanation for Romer's Gap is environmental. The late Devonian extinction and the subsequent changes in atmospheric chemistry, specifically a period of reduced atmospheric oxygen in the earliest Carboniferous, as the decomposition of accumulated organic matter temporarily consumed more oxygen than photosynthesis was producing, created conditions that were hostile to large-bodied air-breathing terrestrial vertebrates. Low oxygen made the air thin in a functional sense. The same volume of air contained less oxygen, making sustained terrestrial activity energetically demanding. The terrestrial vertebrates that made it through Romer's Gap were the ancestors of all subsequent amniotes and amphibians, and the characteristics they carried through this bottleneck: respiratory efficiency, metabolic flexibility, the ability to exploit a range of environments, were the characteristics that selection had refined during 15 million years of challenging conditions. When oxygen levels recovered in the mid-Carboniferous and the coal forests established themselves, the terrestrial vertebrates that emerged from Romer's Gap diversified rapidly into the rich ecological opportunities of the high-oxygen coal forest world. The amphibians, now represented by diverse groups including enormous crocodile-like forms up to 6 meters, about 20 feet, long, that were the apex predators of the Carboniferous freshwater systems, and the first amniotes radiated through the Carboniferous coal forest ecosystem in a secondary explosion comparable in its speed, if not in its scale, to the Cambrian Explosion that had opened the Paleozoic. The Carboniferous rainforest collapse, which occurred approximately 307 million years ago, roughly 40 million years after the coal forests reached their maximum extent, was another event that shaped the terrestrial vertebrate world in ways that resonate through all subsequent evolutionary history. The collapse was triggered by a dramatic shift in climate. The assembly of Pangaea was changing the global circulation of the atmosphere, and the tropical coal swamps that had been continuously wet throughout the early and middle Carboniferous began experiencing more pronounced dry seasons. The continuous tropical forest that had covered most of Euramerica fragmented into isolated patches, separated by seasonally dry habitats that the moisture-dependent lycopsid trees could not cross. For the amphibians, which required wet environments for reproduction, the fragmentation of the coal forests was catastrophic. Amphibian diversity plummeted as their habitat became discontinuous, each forest patch supporting an isolated population that could not exchange individuals with neighboring populations across the dry gaps. For the amniotes, which could reproduce anywhere on dry land and could tolerate the dry seasonal environments between forest patches, the forest fragmentation was an opportunity. The amniotes could cross the dry gaps that the amphibians could not. They could colonize the isolated forest patches before the amphibians could. They could exploit the seasonal environments that the amphibians avoided. The Carboniferous rainforest collapse effectively ended the age of amphibians and initiated the age of reptiles and synapsids that would characterize the Permian. From the first amniotes, two major lineages diverged almost immediately. One lineage led to the reptiles and, through the reptiles, to the dinosaurs, birds, and crocodilians. The other lineage, the synapsids, led to the mammals. And it is the synapsids that dominated the terrestrial world for the first 100 million years of amniote history, before the Permian extinction reshuffled the ecological deck and gave the reptiles their opportunity. The synapsids are distinguished from the other early amniotes by a single opening in the skull behind the eye socket, called the temporal fenestra, where jaw muscles attached. This opening is diagnostic, and it is the fossil character that allows paleontologists to identify synapsids unambiguously even from fragmentary material. In modern mammals, this opening has been enlarged and modified into the complex temporal region of the mammalian skull. But in early synapsids, it was a relatively modest hole in the skull roof. What mattered was not the hole, but what it signified. The synapsids were, from their earliest appearance, evolving toward the enlarged jaw muscles and differentiated teeth that eventually produced the mammalian dental formula. The early Carboniferous and Permian synapsids, called pelycosaurs, were the dominant large terrestrial vertebrates of their time. The most famous pelycosaur is Dimetrodon, which, despite appearing in countless toys and educational materials alongside dinosaurs, was not a dinosaur, was not a reptile in the modern sense, and was in fact more closely related to you than to any dinosaur. Dimetrodon had a spectacular sail on its back, a fan of elongated neural spines connected by skin that was probably used for thermoregulation, acting as a solar collector to warm the animal rapidly in the cool morning air, and perhaps as a radiator to dissipate excess heat during the warmest parts of the day. It was an apex predator of the early Permian terrestrial fauna, with a skull full of differentiated teeth, some canine-like stabbing teeth, some cutting teeth, that gave it a more versatile dietary toolkit than the simple, undifferentiated teeth of most reptiles. Dimetrodon went extinct well before the Permian extinction, replaced by more advanced synapsids, the therapsids, that were moving rapidly in the direction of the mammalian body plan. The therapsids appeared approximately 275 million years ago, and within a few tens of millions of years had diversified into the most ecologically dominant group of large terrestrial vertebrates the world had yet seen. They included herbivores the size of hippos, carnivores the size of bears, and small insectivores whose skulls are so similar to early mammal skulls that the boundary between therapsid and mammal is, in the fossil record, essentially arbitrary. The dicynodonts were the most successful Permian herbivores, two tusked synapsids with a horny beak for cropping vegetation and a highly mobile jaw joint for processing tough plant material. They ranged in size from rat-sized burrowers to Moschops-grade animals the size of a large cow. Lystrosaurus, a dicynodont the size of a pig, would survive the Permian extinction and become the dominant terrestrial vertebrate of the early Triassic world, one of the most dramatic examples of a survivor species, inheriting a world from which most competitors had been eliminated. The Gorgonopsids were the apex predators of the late Permian, large saber-toothed synapsids with elongated canine teeth for dispatching large prey, built along the lines of a modern lion but with enough synapsid characteristics to place them firmly in the lineage leading to mammals. And then there were the cynodonts. The cynodonts are the group of therapsids from which true mammals eventually evolved, and by the late Permian they had accumulated so many mammalian characteristics that the distinction between cynodont and mammal was, for paleontologists, more a matter of convention than of any dramatic biological discontinuity. Late Permian cynodonts had differentiated teeth, incisors, canines, and post-canine teeth with complex cusps for processing food that are essentially identical to early mammal teeth. They had a secondary palate, a shelf of bone separating the nasal passage from the mouth, allowing them to breathe while chewing, a feature absent in reptiles. They had a reduced number of skull bones, with several of the bones that were jaw bones in other synapsids having migrated toward what would eventually become the middle ear bones of mammals. They may have had fur. They may have been endothermic, warm-blooded in the modern mammalian sense. They were, in every practical biological sense, almost mammals, and they were flourishing. The late Permian world, with Pangaea assembled into its final form and the vast interior deserts of the supercontinent providing the harsh selective pressure that drives evolutionary innovation, was a world of extraordinary therapsid diversity. The dicynodonts grazed the floodplains in enormous herds. The Gorgonopsids hunted them. The cynodonts occupied every niche too small for the larger therapsids. The world before the dinosaurs was not the bleak, empty stage that popular imagination sometimes presents. It was a rich, complex, fully realized biosphere built over the preceding 300 million years of Paleozoic evolution, populated by animals that were increasingly sophisticated and increasingly familiar in their biology. And then, approximately 252 million years ago, in what may have been the most catastrophic event in the history of complex animal life. Almost all of it died. The Permian-Triassic extinction, the Great Dying, as it is sometimes called, eliminated approximately 96% of all marine species and 70% of terrestrial vertebrate species. It was not one event, but a cascade of events, triggered primarily by the eruption of the Siberian Traps, one of the largest volcanic events in the past 500 million years, which released CO2, methane, and toxic volcanic gases into the atmosphere over a period of approximately 1 million years. The warming from the CO2 pulse drove ocean temperatures to lethal levels in tropical regions. The acid rain from the sulfur dioxide emissions dissolved the carbonate shells of marine organisms. The hydrogen sulfide produced by anaerobic bacteria in the anoxic deep ocean bubbled into the atmosphere and poisoned terrestrial ecosystems. The ozone layer was destroyed by volcanic halogen emissions, exposing surface life to lethal levels of ultraviolet radiation. It was a perfect storm of catastrophes, and it ended the Paleozoic. The trilobites, which had survived the Ordovician extinction, the Silurian extinctions, the late Devonian extinction, and the lesser extinctions of the Carboniferous, were finally eliminated. They had been declining through the Permian, reduced from their Cambrian and Ordovician dominance to a few specialized lineages, but they had persisted for nearly 300 million years. The Great Dying finished them. The Eurypterids were gone. The rugose and tabulate corals, the reef builders of the Paleozoic, were gone. The brachiopods, which had been the most abundant shellfish of the Paleozoic seas, were reduced from hundreds of species to a handful of survivors. The gorgonopsids were gone. Most of the dicynodonts were gone. The world that had been building for 287 million years was erased in what amounted to, geologically speaking, a moment. What survived was a small fraction of the Paleozoic biosphere. Lystrosaurus, the pig-sized dicynodont, survived and inherited the Triassic world. For a brief time, it constituted roughly 90% of all terrestrial vertebrate individuals on the planet. A monoculture of a single species in a landscape stripped of ecological competition. A few lineages of cynodonts survived, and from them, within the first 10 million years of the Triassic, true mammals would eventually evolve. A few lineages of archosaurian reptiles survived, and from them, within the first 20 million years of the Triassic, the dinosaurs would evolve. The stage had been cleared. The 287 million year opening act of the Phanerozoic was over. What the Iranian sections published in 2026 add to this picture is precision. The kill was not one blow, but a series of pulses, each stripping away another layer of survivors until the cumulative weight of catastrophes exceeded what the Paleozoic biosphere, however resilient, could absorb. But the lava itself was not the primary kill mechanism. The gases released by the eruption, CO2, sulfur dioxide, chlorine, and fluorine compounds, and critically, the methane released when the ascending magma intruded through the thick coal and organic-rich sediment sequences of the Siberian basin, were what changed the world. The CO2 released warming. The warming destabilized methane hydrates on the seafloor, releasing additional methane in a positive feedback. The temperatures in the shallow tropical oceans rose by perhaps 10 to 15 degrees Celsius, about 18 to 27 degrees Fahrenheit, within a geologically brief interval, killing the coral reef communities that had survived every previous Paleozoic extinction. The acid rain from the sulfur dioxide dissolved the calcium carbonate shells of marine organisms before they could reach the sea floor and be preserved, an acid ocean that could not support calcifying organisms. On land, the combination of intense heat in the Pangaean interior, already extreme due to the continental geography, and the disruption of the water cycle by the volcanic emissions created conditions in which large areas of the supercontinent became functionally uninhabitable. The evidence from the Karoo Basin fossil record shows a sharp decline in the diversity of therapsid species in the latest Permian, followed by the near-total absence of fossils for an interval that represents the peak of the extinction. This interval, sometimes called the dead zone, appears to correspond to several 100,000 years, during which the Karoo ecosystems were so severely disrupted that fossilizable bones essentially stopped accumulating. New data published in 2026 from the Aberdare and Baghuk sections of Iran have added nuance to the understanding of the ocean's behavior during the Permian-Triassic transition. The Iranian sections show evidence of oxygenated shallow water conditions in the central Tethys Ocean during most of the late Permian, but with episodic anoxic events that became more frequent and more severe approaching the extinction boundary. This pattern of oscillating oxygenation and anoxia is consistent with a kill mechanism that operated in pulses rather than as a single event, which explains why the terrestrial fossil record shows multiple phases of extinction rather than a single sharp boundary in some regions. The recovery from the Permian extinction was the slowest of any recovery from a mass extinction in the Phanerozoic record. The ecological complexity that had accumulated over 287 million years of Paleozoic evolution was not rebuilt in a few million years. The early Triassic world, the world that Lystrosaurus inherited, was characterized by low diversity, simple food webs, and the dominance of a handful of generalist species. The reef ecosystems that had been a feature of the Paleozoic oceans since the Ordovician did not reform for approximately 8 to 10 million years after the extinction. The terrestrial vertebrate communities of the early Triassic were dominated by a small number of generalist herbivores, the dicynodont survivors, and an equally small number of predators without the complex multi-trophic structure that the Permian therapsid world had developed. The full recovery of ecological complexity took approximately 10 million years. A geological eye blink, but an eternity of generations for the organisms living through it. And the world that recovered was fundamentally different from the world that had existed before the extinction. The Paleozoic fauna, the trilobites, the rugose corals, the large brachiopods, the diverse crinoid communities, the eurypterids, did not recover. They were gone. The marine world that re-diversified in the Triassic and Jurassic was built around different groups. The modern-style corals that eventually produced the modern reef ecosystems, the bivalves and gastropods that replaced the brachiopods as the dominant shellfish, the cephalopods that evolved into the ammonites. The terrestrial world that rebuilt itself in the Triassic was dominated by the archosaurs and their descendants, rather than the synapsids. The dinosaurs replaced the therapsids as the dominant large terrestrial vertebrates. But the synapsids were not entirely displaced. A small lineage of cynodonts survived the Permian extinction and persisted through the Triassic. Small-bodied, nocturnal insectivores that had retreated into the ecological margins exactly as the mammals of the Cretaceous would retreat into the ecological margins under the dominance of the dinosaurs. From these cynodont survivors, within the first 10 million years of the Triassic, the first true mammals evolved. They were small, they were nocturnal, they were ecologically marginal. They would remain so for the next 185 million years until another mass extinction created another opportunity. The story of the Paleozoic ends not with triumph, but with survival. And survival, as the geological record teaches us with unfailing consistency, is the prerequisite for everything that comes next. Now let me tell you what I think about all of this as someone who has spent time with the Paleozoic fossil record. The Paleozoic deserves more than it gets. In the popular imagination, prehistoric life means dinosaurs. Before the dinosaurs, there is a vague awareness of trilobites, those appealing fossils that appear in natural history museum gift shops, and perhaps a general sense of ancient seas and primitive life. But the full scope of the Paleozoic, its duration, its complexity, its extraordinary biological innovations, the grandeur of its ecosystems from the Cambrian Explosion through the coal forests to the Therapsid world, is almost entirely absent from popular understanding. This is a failure of narrative, not of evidence. The evidence is there. The Burgess Shale and the Chengjiang fossil beds of China preserve the Cambrian world in extraordinary detail. The Hunsrück slate of Germany and the Cleveland Shale of Ohio preserve the Devonian world. The coal measures of Pennsylvania and Yorkshire and the Ruhr preserve the Carboniferous world. The Karoo Basin of South Africa preserves the Permian Therapsid world in the most complete terrestrial vertebrate record of the late Paleozoic anywhere on Earth. The fossils are there. The stories they tell are extraordinary. What they tell us, above all, is that the patterns we observe in evolution repeat. The Cambrian Explosion and the recovery from the Permian extinction are fundamentally the same event at different scales. A mass extinction clears the ecological stage. The survivors radiate rapidly into the empty niches, and the resulting biosphere is richer and stranger than what preceded it. The evolution of eyes in the Cambrian and the evolution of endothermy in the Therapsids are both expressions of the same principle. That increasing the sensory and metabolic complexity of an organism opens new ecological possibilities that selection then drives to exploit. The progression from simple microbial mats to Anomalocaris, to Dunkleosteus, to Dimetrodon, to Cynognathus, is not a march toward a predetermined goal. It is a series of contingent accidents, each building on what preceded it, none predictable from first principles, all comprehensible in retrospect. The Paleozoic gave us everything. The trilobite eye is the ancestor of the compound eye of every insect alive today. The lobe-finned fish of the Devonian gave us the four-limbed body plan that every terrestrial vertebrate has used since. The amniotic egg of the Carboniferous gave us the reproductive independence from water that allowed the tetrapods to conquer the dry interior of every continent. The cynodont jaw of the late Permian gave us the differentiated teeth and the secondary palate and the eventual middle ear bones that define the mammalian lineage. Every structure in your body that is specifically characteristic of a mammal traces its evolutionary origin to the Paleozoic synapsids of the Carboniferous and Permian. The coal in the power stations, the limestone in the buildings, the oil in the engines, the oxygen in the air, all Paleozoic. The body plan you inhabit, the teeth you chew with, the ear bones that convert sound waves into neural signals, all Paleozoic. The era that most people have never heard of, that lasted nearly 3 times as long as the age of the dinosaurs, that produced the first forests, and the first bones, and the first eyes, and the first warm blood. That era is, in the most literal biological sense, the foundation of everything alive today. The dinosaurs were spectacular, but they were the sequel. The Paleozoic was the original.